Good morning, everyone. I'm Andrew Hahn, an editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Developing an Integrated MRD Assay to Transform Oncology, and is sponsored by Twist Bioscience. Our speakers today are Bérengère Genin, Chief Operating Officer at IntegraGen, and Charles Marcaillou, Head of R&D at IntegraGen. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Bérengère.
Welcome, everyone, and thank you for joining us today. I'm Bérengère Genin, Chief Operating Officer at IntegraGen. I'm thrilled to be here with our partners at Twist Bioscience to explore how we're helping bring MRD testing into clinical practice across Europe. In the next few minutes, I will give you a brief overview of who we are, what we bring to clinical sequencing, and how we approach liquid biopsy solutions with a focus on MRD detection. Then my colleague, Charles Marcaillou, our head of R&D, will take you through the specific tumor-informed MRD workflow we've developed. To conclude this presentation, I will have the honor of representing Séverine Martin-Laméré, our head of quality at IntegraGen, who unfortunately couldn't join us today, but has prepared valuable clinical insight on how MRD is transforming the oncology landscape in Europe. Let me begin with a short history of IntegraGen. IntegraGen was founded as a spin-off from the French National Genotyping Center and quickly became a key player in next-generation sequencing in France. From our roots in academic research, we've built deep expertise in clinical-grade sequencing with over 15 years of experience in sequencing biological samples, from targeted research studies to large-scale interventional clinical trials. We operate under CLIA and CAP certification and have been the sequencing partner for major academic centers such as Institut Pasteur, Gustave Roussy, and one of the France Medicine Genomic 2025 initiative. We also support biotech and pharma by managing genome data pipelines in regulated environments, including for multi-center clinical trials across Europe. So what exactly do we bring to our partners? Let's take a look. IntegraGen provides NGS services tailored to the needs of academics, hospitals, pharma, and biotech. Our strength lies in delivering high-quality sequencing for both routine and complex clinical projects. With over a decade of experience, we developed a streamlined logistic network across Europe. We are the trusted partner of top-tier hospitals and academic centers and operate under CLIA CAP standards. What makes us unique is our agility, deep bioinformatics integration, and our ability to support demanding turnaround times, even at scale. Let's now take a moment to revisit what liquid biopsy means in the context of oncology. Liquid biopsy allows us to analyze tumor-derived material, such as ctDNA in blood or other body fluids. It is non-invasive, cost-effective, and enables repeated sampling. The key advantage, it captures real-time insights into tumor biology without requiring invasive tissue biopsies, making it highly suitable for longitudinal monitoring. But the real power of liquid biopsy lies in its clinical applications, especially in tracking ctDNA during cancer progression. ctDNA levels reflect tumor burden and can be tracked over time. This graph shows how liquid biopsy enables us to measure response to neoadjuvant therapy, detect minimal residual disease, monitor adjuvant therapy, and detect relapse earlier than imaging. All of this supports more adaptive, timely, and personalized cancer care. Our solutions leverage this potential with high sensitivity and precision, enabling personalized real-time cancer monitoring. Let's now focus on what we've developed at IntegraGen in terms of MRD detection strategies. At IntegraGen, we've built a dual approach to MRD detection. On the left, our tumor-informed MRD test, ultra-specific, based on patient-specific variants from whole exome sequencing and validated using Twist MRD 500 panel. On the right, our methylation-based MRD assay under development with this tumor type informed doesn't require a tissue sample and enables broader application. These strategies allow us to tailor MRD testing for different patient cohorts and trial designs, while maintaining the robustness and specificity expected in clinical settings. Today, we will focus specifically on the tumor-informed strategy, which is the foundation of our clinical MRD offering. And now, to walk you through this tumor-informed MRD workflow in more detail, including the panel design, sequencing, bioinformatics, and performance metrics, I will hand it over to Charles-Marc Alliou, our head of R&D.
Thank you very much, Bérangère. Hello, everyone. So it's now my turn to talk about tumor-informed MRD 500 and dive into our results. So first of all, I wanted to tell you that the presented results could be found in our application note published with Twist. The Twist MRD 500 is a whole solution for tumor-informed minimal residual disease. It's a two-step NGS workflow. The first step is the waste analysis germline tumor from each patient for the selection of tumor variants, the design and ordering of probes for Twist panel production. The green tiles in the workflow represent solution provided by Twist, and blue tiles represent other internal tasks to be done in the lab. 500 is for the scale. 20 to 500 variants or probes could be ordered in one panel large enough to get a relevant personalized MRD panel. For this first step, Twist provides target enrichment products, proven and trusted by the community for several years. The second step is the target enrichment process from plasma DNA samples on these MRD panels for ctDNA detection. It combines the same proven target enrichment Twist solution, and new kits from Twist included the MRD panel with 6 open days TAT delivery for 12 singles or multiplex hybridization, but also the new cell-free DNA library preparation kit with UMI adapters and cell-free DNA pan-cancer reference standard V2 for sensitivity and specificity assessment. Monitoring the performance and reliability of this process was a goal of our work. However, first of all, we can talk about sensitivity considerations in the context of MRD and ctDNA detection. Zviran et al. Modeled the probability of detection of tumor variants in ctDNA as a function of 3 parameters: tumor fraction, input in genome equivalent, and number of mutations captured. The obvious message is increasing the number of mutation markers increases the probability of detecting ctDNA. However, the goal is not to select and find all variants, but to detect ctDNA with a statistically sufficient number of variants. We performed internally another kind of modeling based on the Poisson distribution, the detection probability of variants in a scenario of 42 selected variants. For example, with 20 nanogram, the probability of finding all variants is 0, but optimal to find at least 4 of the 42 at 0.003% BAF. But of course, the ability to obtain this sensitivity is conditioned by the conservation of the input diversity during the process. So for this kind of assessment, we performed a head-to-head comparison of the NGS kits for cell-free DNA library preparation, because it's the step where you could lose the input amount's DNA. We compared the previous Twist MF kit, which already gave solid results, to the new Twist cell-free DNA kit on 6 cell-free DNA samples with 10 or 20 nanogram input. We empirically calculated the library conversion rate, LCR, that is the yield of the library. From nanogram post-PCR produced, number of used PCR cycles, and PCR efficiency, we could assess remaining amounts of the input amounts in the library. You can see that the LCR is optimal with the new kit, enabling optimal sensitivity for MRD assessment. PCR monitoring of the library preparation confirms this observation. Next, we performed a full validation of the whole process tumor-informed MRD 500 with a concrete retrospective study. That's the experimental design that served for the application note published with Twist. DURIGAST validation is a small retrospective study with 8 patients and 3 plasma time points from FFCD, la Fédération Française des Cancers Digestifs. The main 2 goals are our capacity of selection of relevant tumor variants from WES, germline tumor analysis, from FFPE tissue. FFPE rather than frozen tissue is, of course, an additional challenge. And from plasma, a technical validation for LOD assessment, especially from the suitable Twist reference standard V2 that mimics cell-free DNA with known return variants and BAF. And a biological validation with clinical checking of patient cell-free DNA before treatment at V1, after 4 weeks treatment at V2, and after treatment and relapse at V3. For that purpose, we performed 24 libraries from patients' cell-free DNA and 11 libraries from Twist reference samples. That's our primary analysis from WES. Even with FFPE from weak quantity and quality assessed by the DV500, that's the percentage of fragments, more than 500 base pair, roughly the percentage of fragment available for the library. We get reliable NGS metrics in terms of depth and uniformity for variant calling analysis. So now more about analysis and selection of tumor variants. With an updated version of our Mercury analysis tool for relevant tumor variant selection, we use some specific Mercury filters for variant quality and class and database curation that I won't detail. We also perform a curation from clonal hematopoiesis with the screening of variants present in PBMC. However, most of the tumor variants are CTGA artifacts from FFPE. An appropriate threshold of the tumor VAF, the tumor allelic fraction, is chosen to differentiate them from true tumor variants. That's the graph on the right with a typical result. A 7% threshold is chosen in this example, with a red curve corresponding to density of CTGA variants. Blue curve for other variants, and green curve for indels. The threshold allows the elimination of artifact variants, that's the left part of the threshold, and this threshold is specific for each tumor. Finally, from 34 to 119 tumor variants and an average of 78 were designed per patient, even for noisy FFPE tumor. It was therefore easy to combine all found variants for the 8 patients on just 2 panels. So 2 panels for 8 samples, because the study is retrospective and we already collected all plasma samples to be screened. In each panel, we added 82 variance controls, that's the green blocks at the bottom of the left graph. 42 variants for LOD assessment with the TWIST reference standards, and for sample ID tracking and background calculation, 40 polymorphic SNP were added with an average VAF of 50% in European, African, and Eurasian population. Patient plasma final genotypes will be compared with a corresponding tumor germline pair for the ID validation. These 2 panels were ordered from TWIST, and we received the tubes after 8 days in respect of the attended TAT from TWIST. For cell-free DNA analysis, we performed 7 target enrichment captures. Again, because of the retrospective context, we were able to perform multiplex captures on the 2 panels. We performed cell-free DNA libraries from varying input amounts, from 6 nanogram to 30 nanogram, and an average of 20 nanogram. We get optimal library conversion rate for all cell-free DNA with the new library kit, except for patient 6v2. But this library exhibited a red coloration throughout the preparation process with a potential hemoglobin inhibition. A paired N 150 base sequencing was performed on our NovaSeq-X, and we obtained an average of 140 million paired N reads with an optimal specificity of 72% on the targeted probe. Of course, UMI adapters were added in the library prep at the ligation step and allowed tagging of each molecule for a consensus analysis. That's the description of our statistical methodology for ctDNA analysis. For analysis and technical validation, UMI error correction was performed using molecular collapse, following TWIST FG Bio recommendation to get the consensus reads. After consensus, we get a mean base coverage around 6,000x, reflecting the diversity conservation, and UMI family size from 15 to 80 that allowed effective error correction. Into details, we are using SAMTools MPilot for counting each type of base and calculating a VAF. First of all, we are performing this counting for sequencing error determination on the 40 regions designed for sample ID. Outside the center polymorphic SNP, that's about 5,000 positions without variation. Mean error frequency was 2e-5, and there were 3 times more error on CG than on AT when sequencing on the NovaSeq-X. That's our method for background measure on each sample. We perform the same exercise on each designed tumor variant's position and calculated a VAF. On the right, you can see the density of fragments carrying alternative alleles for each base. Blue for the background on the 40 regions, and red for tumor variant's position. Two typical results. On the left, for a sample at a 1.5% VAF. And on the right, a sample at a 0.05% VAF. From this observation, we developed a statistical method of detection based on the binomial law. We compare for each sample VAF associated to background in blue to VAF at tumoral position in red. A variant is considered dissociated from background when the p-value is less than 10 to the minus 3. And to consider a sample positive, we need at least 2 positive variants for a final error probability less than 10 to the minus 6. For the LOD, limit of detection, variant calling and ctDNA quantification were performed using this method. From 20 nanogram input, all twist reference samples with VAF ranging to 0.25% to 0.003% were classified as mutant DNA. An LOD of 0.003% was achieved, enabling the significant detection of 4 tumor variants out of the 42 included in the panel. These results were consistent with the Poisson distribution described in the slide 13. Furthermore, the absence of detection in the wild-type reference sample confirmed the optimal specificity of the test. Now, our final results on clinical samples. On top, the percent VAF for the specific SNVs and in each patient. Below, the percent ctDNA calculated in each patient over time. To conclude, tumor variants were successfully detected in plasma from all patients at all follow-up points, except for a few specific cases. For patient 6, no reliable variant calling could be performed due to the poor tumor DNA quality and technical issues at both V1 and V2. In particular, the V1 plasma sample did not meet our detection threshold, and the V2 sample suffered from low library diversity, likely due to the hemoglobin interference during preparation previously mentioned. For patient 3, at time point V3, the positive sample tracking revealed a mismatch between the plasma and the patient's tumor germline pair, suggesting a handling error before the sample reached the lab. As a result, variant calling was not possible at this time point. Aside from these exceptions, specific SNVs were consistently detected across all other patients and time points. Detected ctDNA levels ranged widely from 0.005% to 26%, aligning with clinical expectations. Higher at diagnosis at V1, lower after treatment at V2, and rising again at relapse at V3. Interestingly, patient 8. It was an outlier, showing increased ctDNA levels at V2 despite treatment, but this was confirmed by droplet digital PCR, suggesting a suboptimal therapeutic response in this case. These results highlight both the robustness of our detection pipeline and the clinical relevance of ctDNA monitoring. Again, the results could be found in the application note published with Twist, and moreover, The whole methodology is detailed in our now accepted publication in JECCR, the Journal of Experimental and Clinical Cancer Research. This paper deals with ovarian cancer and compares this MRD 500 tumor-informed approach with a tumor type-informed approach, also developed thanks to Twist solutions and validated with epigenetic markers, and not requiring prior interrogation of the patient's tumor. That's, of course, another story, but this tumor type-informed approach is at least as sensitive, and once you've pre-selected the epigenetic markers panel, that's not the case for us for ovarian cancer, you don't need tumor tissue anymore for new patients. You can work straight from plasma, and it's much quicker and cheaper to get results. Thank you for your attention, and now I give the floor to Bérangère for the last part of this presentation.
Thank you, Charles, for that deep dive into our tumor-informed MRD workflow and its technical and clinical validation. Now that we've explored the scientific and technical foundation, let's take a step back and consider the broader clinical perspective. To begin, let's look at how ctDNA is changing the game. Circulating DNA offers non-invasive, real-time access to tumor genomics, enabling serial monitoring at multiple time points without the need for repeat biopsies. Because it reflects tumor burden and heterogeneity across all metastatic sites, ctDNA gives a more comprehensive view than tissue biopsy. Its short half-life makes it an ideal tool for real-time disease tracking, especially in a dynamic clinical context. One of its major advantages is the rapid turnaround time of the test, which allows for early treatment initiation when needed. Most importantly, ctDNA can detect microscopic disease that remains invisible to imaging. This is particularly critical in the post-treatment test setting, where minimal residual disease detection can predict relapse risk early, as shown, for example, in post-surgical colorectal cancer. Clinical validity has already been demonstrated through longitudinal cohort studies across several tumor types, including breast, colorectal, and lung cancer. In short, it could give us the flexibility and precision to adapt treatment in real time. However, to transition into routine clinical use, we must now focus on demonstrating clear clinical utility, proving not just that it works, but that it improves patient outcomes. To support clinical implementation, MRD is being evaluated across multiple trial designs, from observational cohorts to randomized interventional studies. First, after curative intent treatment, MRD status can help stratify patients for adjuvant therapy. If ctDNA is detected, meaning the patient is MRD positive, clinicians may escalate therapy to target residual disease more aggressively. On the other hand, if the patient is MRD negative, we can de-escalate treatment and potentially avoid unnecessary toxicity while still maintaining favorable outcomes. This personalized approach may lead to non-inferior survival outcomes with the added benefits of reduced treatment toxicity and improved quality of life. Second, in the post-standard of care setting, MRD testing allows for early detection for relapse. Patients who become MRD positive after completing treatment may benefit from early intervention with additional therapies well before radiological relapse occurs. Those who remain MRD negative can continue with active surveillance with the option to intervene if ctDNA becomes detectable. In both scenarios, ctDNA offers a risk-adapted strategy that supports more efficient and timely treatment decisions, supports smarter use of therapies in trials, and opens the door to a new generation of clinical trials with MRD-guided endpoints. As a result, ctDNA and MRD testing are now transforming the way we design and monitor clinical trials, offering dynamic patient stratification. But challenging remain. One of the key challenges in implementing ctDNA-based MRD testing is ensuring the accuracy and reliability of the result. This begins with standardization, both in pre-analytical and analytical workflows, and in defining threshold for variant detection, especially when we are dealing with low variant allele frequencies. These parameters become critical to reduce ambiguity in interpretation. Accurate detection of low-frequency variant pushes the limits of sensitivity. We are often working near the technical threshold where distinguishing true signal from noise requires a highly optimized assay. Clonal hematopoiesis of indetermined potential, or SHIP, represents another layer of complexity. These age-related or treatment-induced mutations in white blood cells can be misinterpreted as tumor-derived alterations, leading to false positives. This is why matched sequencing of white blood cells alongside tumor-informed approaches is so important. It allows us to subtract SHIP-related noise and focus on true residual disease signals. In the end, the robustness of MRD testing relies not only on the assay technical performance, but also on the strength of the bioinformatics pipeline and the consistency of interpretation. Clear results, appropriate filters, and careful validation of results are all essential to build trust in ctDNA as a clinical tool. The momentum around ctDNA-based MRD testing is clearly accelerating, especially in the context of clinical trials. We are seeing its integration into major studies like Circulate, Dynamic, Umbrella, among others, which are shaping how we evaluate residual disease and personalized post-treatment strategies. This growing interest is also reflecting in evolving recommendations. The ESMO Precision Medicine Working Group, for instance, has started issuing guidance to help harmonize how MRD is incorporated into research and eventually clinical care. What's driving this? The need for more precise, less toxic treatment approaches. MRD is an enabling tool for precision oncology, offering a dynamic and individualized readout of treatment response and risk of relapse. But for ctDNA-guided strategy, to reach clinical implementation, we still need robust prospective evidence. This means large-scale, randomized trials designed to demonstrate not only analytical validity, but clinical utility. Trials that show improved patient outcomes, optimized therapy allocation, or spare toxicity. So while the enthusiasm is real and the adoption is underway, rigorous prospective validation remains the key step to making MRD a standard part of oncology care. Before we conclude, a quick word on quality. To bring ctDNA and MRD testing into clinical practice, we need validated assays with high analytical sensitivity, specificity, and reproducibility. Clearly defined limits of detection are essential, especially when working near the lower boundaries of variant frequencies. But implement standardized, auditable workflows covering everything from sample collection to bioinformatics analysis. For partners, traceability, data integrity, and full documentation are mandatory. A strong quality framework is essential for a reliable clinical tool. To summarize, ctDNA and tumor-informed MRD testing are helping us shape a future where trials are faster, more adaptive, and more personalized. They could support earlier relapse detection, smarter patient selection, and risk-adapted treatment strategies. As adoption grows here in Europe, let's make sure we deliver true clinical value, because at the end of the day, our goal is to ensure that every patient receives the right treatment at the right time. As you've seen today, MRD detection is not just a technological advance, it's a clinical game changer. At IntegraGen, we are proud to contribute to this transformation with robust, sensitive, and personalized solutions validated in clinical settings. Our laboratory operates under CLIA and CAP certifications, ensuring the highest quality standards for clinical-grade sequencing. We combine over a decade of experience with a streamlined logistics network across Europe and an agile infrastructure that supports demanding turnaround time even at scale. What really sets us apart is our deep integration of bioinformatics, enabling real-time data interpretation and customized reporting to meet the needs of pharma, hospitals, and academics partners. We are honored to have been invited by Twist Bioscience to share our journey and to highlight how these partnerships have accelerated innovation and helped bring MRD testing into real-world clinical use. Looking ahead, we are actively expanding the use of our MRD assays across broader patient cohorts and interventional clinical trials to strengthen the evidence base. Thank you for your attention, and please don't hesitate to reach out if you'd like to explore how we can support your clinical or research projects.
All right, thank you, Bérengère and Charles. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. For the Q&A, we'll be joined by Sean Gorda, Principal Product Manager at Twist. First question is for the folks from IntegraGen. Across the 8 patients in your study, how is the number of variants allocated for each patient? And why does it vary between patients?
So maybe I can answer this question. Of course, it depends of the tumor, and regarding, of course, the TMB of each tumor. It's you have this kind of variation across each tumor, and like what you can see on this slide. Yes, you can see the variation and the number of tumor variants that we focused and the one we detected. It's a personalized test, but it's going to vary from each tumor.
You reached a limit of detection of 0.003% from 20 nanograms of input. Have you tried to assess LOD with less than that?
Yes, it's a good question. We performed this experiment after the publication of the white paper. However, if you think about extraction from plasma, from 1 ml, you should extract at least 5 nanogram for healthy subjects, and of course, more for cancer patients. And again, the new results fit with our modeling based on the Poisson distribution and the LCR that is more than 75%. So to be more precise, we got 0.012% for 5 nanogram, and we also get a good sensitivity for 300 picogram of DNA, and the LOD was 0.2% from 300 picograms.
You mentioned one of the assays failing due to sample identity mismatch. Is that a standard control in MRD assays, or is it something that you've chosen as a quality control?
It will be, of course, a standard control, and we're going to put this 40 SNP in the panel. And what I explained in the presentation, it's for sample ID tracking, but it's also to measure the background and to separate the variant tumor from the background. So we need these 40 regions, but it's just 40 regions on a panel where you can put 500 variants, 500 probes.
Why did you use whole exome sequencing for the selection of tumor variants?
Maybe
Bérangère, you want to answer?
Yes, I'm going to take this.
I can do it? Okay.
Yes.
Take it, okay.
No, I will do it. Thank you, Charles. So whole exome sequencing remains the optimal approach for most clinical MRD applications. It offers an excellent balance between genomic coverage and cost effectiveness, while providing sufficient resolution to identify a large number of tumor-specific variants, even from challenging samples such as FFPE. This is critical to ensure robust panel designs for tumor-informed MRD detection, and we've validated our pipeline using whole exome sequencing in combination with a Twist MRD 500 panel. Whole genome sequencing is technically feasible and provides more comprehensive genomic information. However, it is significantly more expensive and requires higher sequencing depths to confidently detect somatic mutations, especially in FFP-derived DNA. In cancers with low tumor mutation and burden, inferior to 1, whole genome sequencing can indeed be considered to ensure sufficient variant yield, but this comes with higher cost and logistical complexity. Alternatively, smaller custom cancer panel could be used, but they must be selected with caution. In low TMB tumors, there's a risk of recovering too few variants, which would limit the sensitivity and performance of the MRD assay. In summary, whole exome sequencing offers a clinically validated, cost-efficient, and scalable solution for MRD panel design with whole genome and custom panels serving as alternatives in specific contexts that require broader or more targeted variant capture.
What was the concentration for the hybrid capture per pool?
Charles, you can take the question, maybe?
I think we're waiting for Charles to refresh his browser. Charles, are you back yet? Sorry, we're experiencing some technical difficulties.
Sorry for that.
We have a question for Twist Bioscience, so for Sean. How would you describe the clinical validation of your MRD tools, and how fast are they getting adopted in the clinic?
We can't really comment on the clinical validation for Twist. It's really about enabling our customers with high quality synthetic DNA to perform the best possible enrichment at a scale and turnaround time that enables a test like this to be clinically valid or clinically possible at scale and at a personalized level. So we're really trying to lower the barrier of entry and unlock this as a possibility for our customers, and like what IntegraGen is doing here.
All right, Charles, do we have you back?
Yeah, sorry for that. I lose the video and the sound. Sorry.
So the question for you was, what was the concentration for the hybrid capture per pool?
Okay, yes, because we perform multiplex captures by 5, and we followed the recommendation of Twist. You have to put more quantity in this multiplex capture. It was, if I remember exactly, 500 nanogram per library in the capture. But that's worked very well, and that's not the step. If you follow this recommendation, you're not going to lose the diversity, because before this step, you have the PCR amplification, and all your copies are inside the capture, and you're not going to lose the diversity and the copies of your material.
All right. How did the use of UMI adapters contribute to error correction and variant calling accuracy?
So, is it for us or for Twist? I tried to explain this in the presentation. Sean, if you want to.
Yeah, I can take this. So the Twist UMI adapters are a set of 32 discrete 5 base pair adapters that allow for 2 things. One is duplicate resolution, and the second is error correction by essentially molecularly tagging library fragments, and then through bioinformatics, comparing the reads to filter out PCR and sequencing artifacts, which allows you to sequence extremely deep and call variants at the very low frequencies demonstrated here.
This is a question for IntegraGen. Where are you in validating the assay prospectively? That question makes sense?
Yes. So do you want that I reply, Charles, on this question?
Well, yes, you can take it.
Or the as you prefer, whatever.
No, you can start.
So
I can add.
Perfect. So about the validation, as Charles explained, for these tests, for tumor-informed MRD 500, the validation of the process is now well established, and the next step for us is to finalize in the next weeks the CLIA/CAP documentation for our work. In terms of prospective, is not a big deal for us. It's the same thing. It's just in terms of designs. If we have a retrospective or prospective study, it will be different, but the validation is exactly the same. It's the same process.
That's a good answer. And of course, for prospective context, in the prospective context, so you have one panel for one sample, and most of the time it's a single hybridization. So the result, the technical result, and the performance will be the same.
This is sort of a big picture question that I think anybody can take a crack at. The adoption of circulating tumor DNA is going to require changes in clinical practice. What do you see as the greatest challenges to ensure accelerated adoption of this technology?
Maybe I can take this point. I think the main challenges is the adoption, as you said in your question. Even if one of the key challenges is implementing ctDNA with ensuring accuracy, reliability of the results, we have to standardize both pre-analytical and analytical workflows and avoid any ship and so on. But my point of view is now to convince and to adopt this kind of practice. First of all, in clinical trial, and then in current practice.
Anybody else want to chime in on that, or are we good?
No, it's okay for me.
All right. Looks like we have another question for Twist. The attendee says they noticed the Twist MRD Rapid 500 panel provides sufficient material for 12 tests. Could that be scaled up, or would it require a separate order?
For the off-the-shelf offering, it would require a separate order, but we can customize depending on how many time points a test provider is trying to bring online. At the time, 12 we believe is sufficient for what has been proven and what the field is doing now. But that can be scaled up depending on need.
How do you approach subclonal mutations, particularly in the context of development of resistance mechanisms, which are mainly responsible for cancer relapse?
So maybe this one is for me. About subclones, generally, subclone will retain the majority of mutation identified in the dominant clones. So therefore, we can still detect the presence of ctDNA. However, identifying new acquired subclonal mutations remains a challenge. In our case, the main objective is to assess the presence of minimal residual disease, and we do not consider clonal expansion to be a major issue in this context.
As a reminder to the audience, if you do have any more questions, please enter them into the Q&A chat box. How is the circulating tumor DNA proportion calculated?
Yeah, I didn't precise at this point in the presentation. ctDNA quantification using the tumor-informed approach was performed by calculating the ratio of sequencing reads carrying tumor SNVs to the total number of reads overlapping the genomic coordinates of all tumor SNVs identified in the patient's tumor tissue. So that's how we calculated the ctDNA proportion. But be careful, it's not a direct calculation of tumor fraction. If we want to calculate the tumor fraction, it will be different. It's more complicated because we would have to consider tumor VAF, do a fold 2 for heterozygotes, and find the clever formulas for subclone. In fact, it depends on what you want to quantify. But for ctDNA proportion, it's easy.
All right, well, it looks like that's all the questions that we have for today. We'd like to thank Bérengère Genin, Charles Marcaillou, and Sean Gorda, as well as our sponsor, Twist Bioscience.
Thank you very much.
If you had any other questions, please submit them and we will try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar and would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this GenomeWeb webinar.